Okay, here we go. What is to follow is a review for Anatomy and Physiology 1, the lab final exam. Okay, lab final exam. And as with all lab examinations, I would personally consider this test not to be particularly difficult. If it was multiple choice, you'd all make A's. But what makes it tougher is the fact that it is, in fact, fill in the blank. So, as a fill in the blank test, it comes with an added layer of complication. That is to say that you have to really know what you're talking about, or else you will have no earthly idea how to answer the questions. So, if you can't look at my lab quizzes as an example and know the answers without having to see the multiple choice options, you are not prepared for this test. Not prepared. Uh, a wonderful way of getting at this is to take my PowerPoints and go through the muscles on the body as an example and remove the words, remove the words. And as I go through and I'm pointing at things on the video, you should see if you know the answers without me having to call it out. Like you can even put the my lab video on mute. And as I'm pointing at things on a model, if you can't tell me what those things are I'm pointing at, you are not prepared for my exam. So just throwing it out there for you. This is the way of things. Yeah, there we are. All right, here we go. I'm going to go ahead and review you for this thing, and we'll see what happens. Hopefully, I don't get attacked by children. We will see. All right. Part 1, muscles. Oh my gosh. You can watch the video and probably tell which muscles I like more than others. Hey, I got a question. Am I too echoey in here? Do I sound, Landry, do I sound pretty normal?
Yeah, you sound normal to me.
Okay, I just don't like it. Okay, here we go. Here we go. You can probably tell which muscles I like more than others. So like chest muscles, pectoralis major, pectoralis minor, abdominal obliques, rectus abdominis. Like I love those things. Okay, love those things. Your serratus anteriors compared to your rhomboids or antagonistic pairings. Your biceps compared to your triceps or antagonistic pairings. Basically, any muscle that I gave you an origin and an insertion and an action for, those are pretty much all on the test. All right. I did not give you a great number of origin, insertion, and actions, but the ones that I did give you, they are all going to be on your exam. So I would come prepared with that in mind. All right. Part 2 of this conversation. Think about your deltoids as lifting your arms, that is abduction. Think about your latissimus dorsi muscles as pulling your arms down. That would be abduction. I don't know why I'm displaying all this for you guys. You can't even see anything but my head here because of this camera, but that's life. Let's see what else I got for you. Arms. Oh, what are flexors? What are extensors? Like my rectus abdominis muscles would be flexors. They're pulling me down. My back muscles, like my scalenes in my neck, would be extensors. Here, these are flexors in my forearm. Back here, these are extensors in my forearm. Like there are ways of me getting at a bunch of these concepts that you should be familiar with. All right. So yeah, indeed. Like I might ask you what muscles you'd use to lift your toes up. That would be like your tibialis anteriors. I could ask you what muscles you'd use to point your toes. That would be like a gastrocnemius. Like there are ways of getting at these things, and you should be familiar with these ways. Yeah, I like that. That ain't bad. Okay, I'm flipping through my notes here, team. See what else I've got to say. All right, that'll do for time being. Let's see. Oh, I might ask you some parts of a knee joint. So lateral collateral, medial collateral, anterior cruciate, posterior cruciate. I might ask you, let's see, maybe a patellar ligament. I might ask you what meniscus is. Right? Those are all fair game. I love like the action of the sartorius. I love parts of the quads. That would be vastus lateralis, vastus medialis, vastus intermedius, rectus femoris. I might ask you about the action of the rectus femoris. Like these are all good, fair game items and things that you should probably keep in mind. Yep. All right. I'm working on it, team. Give me a second. Okay. Adduction, or oh, I will definitely ask you some origins and insertions. I might ask you about like if I was lifting something up with my arm, what's a prime mover, like a bicep? What is a synergist, like your brachialis muscle? What is a fixator, like your rhomboids in your back? What is an antagonist, like your tricep? What are they doing for you in reality? And I might take you down the arm and give you brachialis compared to brachioradialis. I think those are fascinating also. And that should be pretty much every muscle I would ask. We'll see. But yeah, I think I've given you a pretty good review on muscles. Again, if you look at the video where I walk you through what muscles are what, I make it pretty plain which ones you should know. So there you go. All right. On to the nervous system. Look, I love the cross-sectional view of the brain. Okay? I love the cross-sectional view of the brain. I love to see like corpus callosum and fornix. And when you crack open the corpus callosum and the fornix, you can see into a lateral ventricle. So grasp that that is in fact a lateral ventricle. Tell me that that would contain cerebrospinal fluid, this sort of thing. Okay. What is a choroid plexus that would make cerebrospinal fluid? So corpus callosum and fornix, the pons, the medulla oblongata, being able to label the cerebellum and the arbor vitae within the cerebellum, uh, labeling the pituitary gland, maybe a pineal gland. Yeah, those are all fair game for this exam. Corpus callosum, yeah, choroid plexus, pons, yes, hypothalamic pathways, yeah, the hypothalamus for sure. And I might even ask you a function for the hypothalamus. Like, if you don't know that at this stage, we got our own problems. So just be ready. And it is not uncommon for me to ask you about lobes of the brain on this test, like putting a star on an area and saying, what lobe of the brain is this? And what does it do? Okay, frontal lobe, oh, it's conscious thought. Occipital lobe, that's vision. You get my point? Yeah, man, it's coming. It's coming. So in particular, think about where does hearing get interpreted in the brain? Where's vision get interpreted in the brain? Where does taste and smell? Like just think about these major things. Because what I would love to do is tie the lobes of the brain in to your what do you call it? Cranial nerves. I would love to tie that into your cranial nerves. So you need to be able to look at a sheep brain dissection and label the first 6 cranial nerves by name: olfactory, optic, oculomotor, trochlear, trigeminal adducens. You need to be able to tell me a little something about all 12 cranial nerves. Okay, label the first 6 off the brain, and then tell me a little something about all 12. And what I need is this: you got to know all their names. And I trust that you guys, you may not have yet. If you go to Canvas and you look at the lab worksheets, you will find that I have provided you with a beautiful worksheet that tells you all the cranial nerve names, how to learn them, what type of nerve they are, and a very, very simple explanation of what they do. And what I try to do is I try to stick to the nerves that matter. Like some of them are pretty obscure in how they function, but like your vestibulocochlear nerve, that's going to play a role in your hearing and equilibrium, which is literally a focus of a whole section of this lab and lecture period. So knowing everything there is to know about a vestibulocochlear nerve, totally fair game. Um, Anything glossae means tongue, so like glossopharyngeal. Facial, you get a facial nerve, so realizing it controls muscles in your face. Like I try to stick to the more straightforward ones. And again, you got to know the name of the nerve, what type of nerve it is, and kind of a little something about what it does. And when I say what type of nerve it is, what I'm asking is sensory, motor, or both. It's either a sensory nerve, a motor nerve, or it has a mixed function. I would call that both. So, like for instance, what is your first cranial nerve? Does anybody know offhand? Somebody make me happy. Cranial nerve 1, nobody? Probably ain't gotten there yet, huh?
Is it ocular?
Not yet. Olfactory.
Olfactory. I knew
Yeah,
it was an O.
I got you. So you have an olfactory nerve, olfactory, optic, oculomotor, trochlear, trigeminal abducens. This is the first six. Olfactory, take a wild guess what that helps you with. Your sense of
Is it
what's
smell?
olfaction?
Smell?
Yup. So that's your sense of smell. And then, of course, you've got optic. That would be
I don't want to
optic.
answer them all, but it's vision.
Yeah. So think about olfaction. Olfaction is a sensory nerve. Okay, it's just information coming in for smell. Optic, sensory nerve, information coming in from your eyes. Then oculomotor is a motor nerve, and it's sending sending impulses out that let me move my eyes around and control my pupillary diameter. So that's just what you got to think about. All right, that's how this is going to go. Okay, quick rehash of what we just discussed. So you know you've got your internal brain structures on a cross section. You know you've got your first six cranial nerves off of a brain that you got to see and identify. You know you got to know about all 12 off of that sheet that I gave you. It is on Canvas right now. Go and look at it. It's beautiful. I made it just for you. It's great. We know about the lobes of the brain and kind of what they do. All right, couple more last but not least situations. You need to know about the spinal cord model. Okay, be able to label the gray matter versus the white matter in the spinal cord. What is a gray commissure? What's a central canal? And what flows through a central canal? All right, team. Choroid plexus. What does a choroid plexus make? Somebody that isn't Kelly.
This might be wrong, but is that where the CSF is made?
Of course. So cerebrospinal fluid gets made in the choroid plexus. It is inside the brain in those ventricles, like the lateral ventricles between the corpus callosum and the fornix, and it flows all the way down the spinal cord through that central canal. This is all very much meaningful and something you need to be keeping in mind as we go. Yep, that's how that works. All right, bear with me here. All right, that's good. That's good. All right. Oh, and on the spinal cord, dorsal root ganglions versus ventral roots. Just kind of think about the orientation of the spinal cord. We should be good on that. Yeah, I like it. All right, this is last but not least, team. Last but not least, senses. And this is a lab that you have definitely not done yet, but this will prepare you to do it this upcoming week. So away we go. Oh my gosh. I put so much on this test about the eyes and the ears. It's coming. Okay. So think about your cornea and your lens. Be able to label the cornea compared to the lens. Tell me about how the cornea is the primary light bending apparatus of the eye. So the cornea really drives what vision is for you, whereas the lens just kind of fine tunes your vision. Tell me about the orientation of the retina. Oh my gosh. I will be putting a what do you call it? A micrograph, so an image from a microscope image on this test of the retina. And you got to label the ganglion cells that look kind of round like this coaster. Hey, they look kind of round ganglion cells. And then those will be connected to bipolar neurons. And then the bipolar neurons are connected to rods and cones. Those are photoreceptors. And then those embed in this very dark melanin pigmented dark structure called the choroid, which basically provides the nutrient supply for the retina. Yeah, man. So ganglion cells, bipolar neurons, rods and cones, choroid. I would label those in order. I would know that in that situation, light would be coming from this way and going that way. It's got to go through the ganglion cells, through the bipolar neurons before it eventually gets to the photoreceptors that are in fact rods and cones. That is on this test. I would be prepared. I would even be able to use terms like transduction. Transduce means to like transform one thing into something else. So your photoreceptors, as an example, transduce light into action potentials. And I think that's a meaningful concept. Yeah. Yeah, it works just fine. All right, let me see what else I got. All right. On the ear, oh my goodness. You'd better be the best of friends with the cochlea, which houses the organ of corti, and that deals with hearing. The semicircular canals, which house the cristae ampullari, and how they deal with cristae ampullari that deal with your equilibrium sense of rotation, and your vestibule that deals with your linear movements and contains a neural structure called the macula. Yup, that's going to be on this test, so be prepared. Be prepared. All right, let me see what else I got. Photoreceptors, microscopy, choroid, ciliary body, lens. Oh, realize that the lens fits inside of a muscular structure called the ciliary body, and it has the ability to squeeze that lens or stretch that lens depending upon what is necessary for your vision processing. So it is entirely possible that I could give you a picture on this lab final of the inside of an eye with a lens sitting in a ciliary body and saying, what is that? The lens. What is that? The ciliary body. What is their relationship? And that is that the ciliary body is a muscle that can squeeze or stretch the lens to help you fine tune your vision. I think that's a really neat way of getting at a fascinating problem. So be prepared. All right. We are pretty much at last but not least here. So in a model of the ear, you will be able to see, let's see here. You will be able to see the external ear, the lobule, the helix, the pinna, whatever it's called in your book, the auditory canal, and that will lead up to a tympanic membrane. Look, your tympanic membrane, you would often call what? What do you call it? Your ear.
Eardrum.
Yep. This is one of the most important parts of hearing. It's probably going to be labeled on your test. And it's really important towards what we would call conductive hearing loss as well. So if you're going into a medical field, having a grasp for this is important. So be able to label the tympanic membrane for me. Be able to label the three ossicles. That would be the malleus, incus, and stapes. You got to know them in order. Malleus, incus, stapes. The malleus is kind of connected to the tympanic membrane. The stapes is attached to the cochlea, the structure called the oval window. You need to be ready for this. This is gonna be on your test in a big way. So be ready. Let's see what else. I've already talked about the cochlea and the organ of corti. I've already talked about the semicircular canals and the cristae ampullaris. I've already talked about the vestibule and the macula. Those are absolutely on the test. Vestibule, linear movement, forward, backward, right, left, up, down. Semicircular canals, rotational movement, equilibrium. And the cochlea, obviously, organ of corti dealing with hearing. I've already talked about the ossicles and the names of the ossicles. I've already talked about the vestibulocochlear nerve that you should be best friends with. Be able to label the vestibulocochlear nerve off of the model of the ear. Be able to tell me if it's sensory motor or both and give me an idea what it does. Yep. And types of hearing loss. So if there's something that always surprises me, it's students missing the types of hearing loss, and it drives me crazy. There are two types of hearing loss. All you got to write is either sensory neural or conductive. That's it. And I'm going to put 3 questions on this test, and all you got to write is sensory neural or conductive. And it is not hard to know which ones are which, but people miss it all the time. So I'm going to give you the dime tour, Lantry. Here's how you do this. If it's conductive hearing loss, the sound waves cannot get to the cochlea. That's it. So, Kelly, if my eardrum's busted and I can't hear, what kind of hearing loss do I have?
Conductive.
It's conductive hearing loss. Okay, I can't get the sound waves in. Maybe I've got a genetic malformation and my malleus, incus, and stapes are fused together. Like, it could be anything, folks. It could be anything. So, conductive hearing loss. For whatever reason, You're not getting sound waves into the cochlea. Let's see what else. And then, of course, there's sensory neural hearing loss. In sensory neural hearing loss, the problem is the cochlea or the nerve coming off the cochlea. Do you have the hair cells in your organ of corti? Are they destroyed? No. Is your organ of corti damaged in some way? Were you in a car wreck and your vestibulocochlear nerve was damaged? Well, guess what? You have sensory neural hearing loss. Okay? That's what that is. It's a neural problem and it's harder to fix. And I feel like that is a pretty decent walkthrough of everything I would study for your lab file. Does anyone have questions for me?
Is there anything about taste or like the tongue on this? No, okay.
It was omitted from your lab manual for some reason. So I end up just not using it, which breaks my little heart a little bit. That's fine.
It's on our lab thing, so we'll
I
learn
know.
it anyways.
I know.
We'll learn it.
It's in your lecture final, so I don't feel as bad. All right. Yeah, it's tough. If it was up to me, I would teach a lot more material, but you guys would hate me more than you do already, so I don't. I've been trying to bring it down a little bit, and that's one of the things that I don't do as much of in lab anymore. Anybody else?
I don't know if maybe you covered this or posted it, but are you gonna post the other 2 spots to turn in for the dissections?
I actually have a sticky note on my desk right now saying to post those. Would you do me a solid and message me on Canvas again? You might have already done it. Somebody
Yeah,
did.
absolutely, I will.
Yeah, that's me on Canvas, and I'll put it up. Like today was pandemonium, and it's on my note as a sticky note. But if you'll send me a Canvas message, that'll be 2 identifications. Because all this is going to do is give you some extra points, which I'm totally down with. So, yeah.
Sounds good, thanks.
Sure. Anybody else? All right, this is what I'm going to do. Hello and welcome to a review for Anatomy and Physiology 2, Lab Final. Okay, Lab Final. You've heard me say this before, and I'm going to say it again, and you probably hate when I say it, but the truth is, this is not a hard test. It's just the fact that it's fill in the blank. If it wasn't fill in the blank, if it was multiple choice, all of you guys would have A's and B's. No question about it. But the nature of it being fill in the blank makes it tougher. So if you can go through my quizzes and answer the questions on the quizzes without having to have the multiple choice, you're probably making big strides towards being successful on my exam. Okay. All right, here we go. So I'm going to start with respiration and go on from there. But before I start on respiration, I want to give you 3 little gifts. Okay, hint, hint, nudge, nudge. These are things that I would be best friends with if I were you. Gee, I don't know. Maybe you could write me a little paragraph about each one. Miss Mack, you with me? She might be. Okay, here we go. All right. I want to know about your liver and bile. I want to know the ducts through which bile flows. I want to know about the gallbladder storing bile. I want to know about the duct that bile takes from the gallbladder. I want to know where that duct goes. I want to know where bile ends up. And I want to know why it goes there. All right. I want to know everything there is to know about bile. There we go. Where does it come from? Where do you store it? What are the names of the ducts it travels from? Where does it go? And what does it do there? That's what I need. All right. Part 2. I want to know about the nephron. Oh my gosh. I want to know about the nephron. I want to know about your glomerulus. I want to know about your proximal convoluted tubule, your loop of Henle, the distal convoluted tubule. I want to know about your collecting duct. I want to know about glomerular filtrate and sort of what's included there. I want to know about how the loop of Henle dips down into a saltier environment. I want to know about how the collecting duct pulls water out of What will be urine eventually? Yeah, man, I want to know all about the nephron. Everything there is to know about the nephron. You should be able to tell me everything there is to know about the nephron. And then last but not least, I can't read my own writing. Oh, geez. Yeah, obviously. I make a really big deal out of us basically all having the same parts and pieces in terms of reproductive structure. Because we do. Up until about week 8 of development, we have the same parts. But then hormonal variances kick in and things change a little bit. Okay. So that is to say, you need to be able to sort of compare and contrast male and female reproductive parts about what structures are analogous to others. So what do we have that is quite similar to our counterparts? And the answer is just about everything we've got has analogous structures to our counterparts. I need you to realize that. I think that's fascinating. I think it's a good way to think about our systems and development. That's what I want from you. Question? Okay, here we go. So now I'm going to dive right in and we will do the rest of this review and we can hope for the best. That bird's not drinking Celsius.
Now you're drinking sparkling water. Just sparkling.
Indeed. All right, here we go. Maybe it's because I'm jealous and I want the Celsius right now in a big way.
I could use a Celsius.
Yeah, I'm telling you. All right, here we go. Respiratory tract. Oh my gosh, the respiratory tract. How many times do I have to say, boy, you should be able to walk me through walk me through the respiratory tract. What's included in the upper respiratory tract? What's included in the lower respiratory tract? Tell me about how your nasopharynx has a very different epithelial lining compared to your oropharynx. Okay. Tell me about how your oropharynx has a very different epithelial lining compared to your tracheal passages. Oh yeah, like I want to know, where would you find pseudostratified ciliated columnar epithelium? Where would you find stratified squamous columnar epithelium? I want to know what those linings look like. Be able to walk me through the hard palate and soft palate. Tell me what the uvula is. Tell me about your epiglottis and how it works and label that thing. Tell me about the glottis and your vocal folds. Oh, love it so much. Oh, I just love this stuff, man. Tell me about your thyroid cartilage and your cricoid cartilage and your arytenoid cartilage. Tell me about how your tracheal passages have little C-shaped cartilaginous rings that keep your trachea open when you're putting it under negative pressure. Tell me about how your diaphragm generates the negative pressure that allows you to breathe. That's amazing. Tell me about how your liver, not your liver, unless you got problems. Tell me about how your lungs have lobes. Yes. Tell me about how your lungs have lobes. I will absolutely make you label trachea and then primary bronchi, secondary, tertiary, all the way down to your bronchioles. Tell me about your alveoli and how they are simple squamous epithelium. Tell me about what the groups of alveoli are called. Oh, tell me about the capillaries that are going, or the artery and vein, let's call it artery veins, that are going to and from your alveoli. Remember, team, this is pulmonary circuit. The definition of an artery is it's coming from the heart to a structure. So in the alveoli, the artery coming in is carrying deoxygenated blood, whereas the vein coming off is carrying oxygenated blood. That's really weird.
Yeah.
That's right. That's right. Who's an old man? Who's a good-looking old man? That's right. He's a good-looking old man. He says, I'm a little gray, but so is my daddy. Indeed. Is this like a pet meeting at this stage? Miss Mott, you should feel a little left out, I think. Oh! Oh, man. All right. Where am I? Because I totally haven't even looked at my. Oh my goodness gracious! Look at us. All right, I haven't even looked at my study sheet. I've just been talking. I hope it's been good. Okay, let me just read a little bit. What are the linings like in the trachea, primary bronchi, secondary bronchi? Explain the diaphragm and breathing. Structures of tertiary bronchioles, alveoli, blood vessels, arteries compared to veins, oxygenated versus deoxygenated blood. Yup. Hard palate, soft palate, uvula, glottis, epiglottis, thyroid cartilage, vocal folds, etc., etc. Okay, I think we're on digestion. Okay, here we go. For digestion, take me top to bottom. Walk me through the mouth. What are salivary glands doing for you? Tell me about how your esophagus runs beside your trachea, but it's a muscular, non-reinforced tube, and it's capable of peristalsis. Walk me down to the stomach and tell me about the inner lining of the stomach. What are rugae? Tell me about the parts of the stomach, like the fundus and the pylorus. Walk me through the sphincters that are there and kind of what they do, like why you have them. So, like the upper sphincter on the stomach, if that wasn't working well, that would cause you to have all kind of heart problems, heartburn, like it would hurt your esophagus. Yeah, not your heart physically, unless you got a major problem. Yeah, that's good. Peristalsis. Yep. Okay, parts of the small intestine and kind of what happens where. Where would you find Peyer's patches and Brunner's glands? Parts of the large intestine. So, ascending colon, transverse colon, descending colon, sigmoid colon, rectum, sphincters therein, like all of that good stuff. Man, I want to know all about it. I want to know all about it. In other words, yet again, how many times do I have to say this? I will give you a figure. You're going to take me top to bottom through that digestive system. Okay, now on to task. I will absolutely put an image on this test. I probably have made you guys a study guide of the images, so you can go through and find this one. It's going to be where I've got the liver and the pancreas and the ducts that connect them together, and the stomach and the large intestine, and sort of how all of that connects in with one another. And you got to label a lot of parts, okay? Especially like the gallbladder and like all the ducts coming off of that. Cystic duct, common bile duct. I want to know all about it. All right, I want to know all about it. And yet again, tell me what bile does for you, all that fun stuff. Yeah, that's going to happen. That's going to happen. Good. Pancreas, duodenum, good. I will probably put a pancreas slide on this test and make you label islets of Langerhans. Okay, and give me a little detail about what's happening where. I will probably put a liver slide on this test. You'll have to label like a portal triad, or a lobule, or a hepatic portal vein, or some kind of ridiculous something. Okay, so a little bit of microscope slide action, definitely a pancreas, definitely a liver. And while I'm thinking about microscope slides, I will absolutely put a glomerulus on this test and make you tell me this is a urinary system in the kidney, and this is a urinary production center. I will absolutely put an ovary and a testis on this test on microscope slides. On the ovary, you'll be labeling like a primordial follicle or a primary or a secondary or a Graafian follicle, maybe an oocyte. And on the testis slide, you'll be labeling maybe Leydig cells and tell me they make testosterone. You'll be labeling a seminiferous tubule, be labeling some sperm cells or their tails, anything along those lines. All right, major functional parts. That's what I'm going for here, folks. Major functional parts. I feel like that's the only microscope slides that come to mind for this particular test. Maybe making you look at Peyer's patches and telling me there's Peyer's patches that they, whatever, where they are in the intestines. Yeah, that might work. Okay, on to the urinary system. Oh, shoot, I got two more. All right, on the urinary system. So on a kidney, label the whole darn thing for me, okay? So cortex versus medulla. Do the blood vessels. renals, interlobars, corticoradiata, like all that good stuff. Arcuate arteries. What else? So out in the cortex is where you find the nephrons, and I've already told you, you got to be able to label all the parts of a nephron: glomerulus, proximal convoluted tubule, loop of Henle, distal convoluted tubule, collecting duct. Tell me what the collecting duct does as it runs through that salty environment, pulling fluids out of what would become urine. Tell me about the renal pyramids and their stringy nature as a result of those collecting ducts. Tell me about how that's a really salty environment. The pyramids lead to minor calyxes. Minor calyxes lead to major calyxes. Major calyxes lead to the renal pelvis. Renal pelvis leads to the ureter. Ureter leads to the bladder. Bladder leads to the urethra. Urethra leads out of the body. Tell me about how you have a skeletal muscle-based sphincter and a smooth muscle-based sphincter on the urethra, and how that allows us to control urination. That's great stuff. Let's see. Segmental, interlobar, arcuate, cortical radiata in the kidney. Yes. Renal pelvis, ureters. The ureters are, whatever you call it, they are muscular. Yep. Okay, what else we got? Okay, I believe that takes us down to reproduction. All right, I've already walked you through the microscope slides for reproduction. So there will definitely be an ovary slide. There will definitely be a, whatever the other one is. I don't know which one I said first. It's an ovary and testis.
Yeah.
You get my point. So one or the other, it's going to be on, or both of them will definitely be on there. And then, of course, I will give you a cross-sectional view of the male reproductive system. I will give you a cross-sectional view of the female reproductive system. And I'll give you like 3 or 4 things off of each of them that you have to identify. That is gonna happen. So yeah, I will stick to major things. Testis, ovaries, fimbriae, epididymis, maybe like a prostate gland. I won't put something itty bitty tiny, you know. It's not. I'm not gonna put a bulbourethral gland on there. You know, you get my point. Like, I will try to stick to serious structures. You know, cervix, big things that matter that you're gonna be dealing with as a nurse, as a medical professional. So yeah, that's how that's gonna work. That's how that's gonna work. And there may be questions on there. I might ask you, nah, probably not. I might ask you something about where sperm cells are made compared to where they mature, because it's kind of different than an ovary. And I think that's fascinating. So just keep that in mind as we do what we do. Ooh, or alternatively, I might ask you where fertilization might take place, like not in an ovary. Okay, it's an fallopian tube. So just functional aspects, like something you're like, oh yeah, that's how that works. And team, I'm telling you, man, I think that's pretty well it. All right. Does anyone have any questions for me?